All right, welcome back. So this is going to be part 5 in our excitable cells lecture. And what we'll do here is we'll begin talking about something known as a graded potential. We'll look at the structure of our neuron, and then we'll begin to get into the actual mechanics of the action potential. So this idea of a synaptic potential being graded. And so what we'll look at in a second, we'll look at what a synapse is. A synapse is essentially where two neurons meet. So the end of one neuron and the beginning of the other neuron, where this neural signal, where this electrical signal, the action potential is being transmitted and relayed from one cell to the next. That's a synapse. And so the synaptic potential can be graded. And so if you think of the term graded, like grades in school, or grades on a paper, there are different degrees of grades, right? So you can have a very good grade, you can have an average grade, you can have a very poor grade, right? So graded potentials come in different sizes. There are very large, positive, excitatory graded potentials, and there are ones that are very negative and inhibitory. And so we'll look at these different types here, all right? So what this graph is showing is, on the top it's showing the different types of stimuli, so a particular stimulus that's arriving at a neuron, and then below you have the corresponding change in the membrane potential. All right, and you can see that a particular type of stimulus causes a similar type of change in the membrane potential. So what this means is that if you have a stimulus that is inhibitory, it is a negative stimulus. So in other words, it's putting us and moving us further away from getting our action potential. What you see is these three right here. So these are both, or these three here are negative, inhibitory, or what we'll call hyperpolarizing stimuli, all right? So what they cause, these hyperpolarizing stimuli, is they cause a movement of the membrane potential in a more negative direction, all right? So the reason that this is a hyperpolarizing, so if you think of the term polar means opposite, right? So you have a positive and a negative pole of a battery, or a positive and negative pole of a magnet, or the north and south pole of the earth, right? Assuming that the earth is round, right? Which it's round. And so you have a top and a bottom, right? So if you hyperpolarize something, you exaggerate the polarity, right? You make those two things further from each other. So if you think of our cell membrane is polarized, right? It's negative on the inside relative to the outside. And so a hyperpolarizing stimulus makes the inside of the cell more negative, and so thus moves the two sides of the membrane in terms of the voltage, moves it further apart. So a hyperpolarizing stimulus of this magnitude causes a hyperpolarization of the membrane of this magnitude, right? So you can see that as you have a larger hyperpolarizing stimulus, or a larger negative or larger inhibitory stimulus, you have a greater decrease in the membrane potential, right? It goes more negative. Now, the reason that this is, we're also calling this negative or inhibitory, is because it makes it more difficult for us to get an action potential, right? And so in order to get the action potential, you have to get your membrane to this dotted line here known as threshold, all right? This graph is showing threshold at about -50. We'll talk about it being somewhere around -55 or so, okay? So obviously, if we move further away from the -55 or -50 in this case, so we are at -70 and we go to -75, right? That's moving further away. So we hyperpolarize, we increase the difference, right? It was -70, now it's -75, -80, so on. And so that moves us further away from threshold, so it makes it more difficult to reach threshold and thus have an action potential. So this is why we're calling this hyperpolarizing or inhibitory or negative. All right, any and all of those terms are meaning essentially the same thing. Okay. Now, if you go over to the depolarizing side, so now we look at these three types of stimuli. You see that they're in the opposite direction. Okay, so you have a small depolarizing stimulus, a little bit larger one, and then an even larger one that corresponds to a slight depolarization of the membrane, a little bit greater depolarization of the membrane, and then ultimately to the point where you reach threshold, and then you can have an actual action potential. All right. So if you depolarize to something, you are taking away the polarity. You are decreasing the polarity. So in other words, you're moving closer to zero. All right. You're moving closer to threshold. You're becoming more positive. All right. So you could call a depolarizing stimulus an excitatory stimulus or a positive stimulus. All right, so in other words, we're moving closer to threshold. Now, you see with our depolarizing stimuli, we have two categories. You have subthreshold and you have suprathreshold. What this is talking about is, does that particular stimulus cause your membrane potential to reach threshold, reach that negative 55 or so? So you can see that the first two are subthreshold. This stimulus here and this stimulus here do not cause the membrane potential to reach our negative 55, right? However, so these are subthreshold. However, our suprathreshold causes our membrane potential to reach threshold, and then if we reach threshold, we get an action potential, all right? Now, it turns out that there's actually an extra step, and that if we reach threshold, we do get an action potential, but only if we reach threshold at a particular point along the neuron, right? And we'll look at that in just a second. This region is going to be known as the axon hillock. If you reach threshold at the axon hillock, you'll have an action potential. If you're not at threshold at a region called the axon hillock, then you will not get an action potential, all right? And we'll talk about that in a second, okay? So this graded potential. They are graded, so you obviously can see here that they come in various gradations, right? There's very small ones, there's even larger ones. This is occurring at the region known as the synapse, which is this general region here. So you have a cell here, so the end of one cell and the beginning of another. In this kind of whole region, this is referred to as the synapse, all right? Now, at that synapse, we release different types of neurotransmitters. It may be acetylcholine, it may be something known as GABA. There could be various types of neurotransmitters. And those different types of neurotransmitters and the type of receptor that they bind to will have some effect on that postsynaptic cell. So it will either be excitatory, it will be a depolarizing stimulus, or it will be inhibitory. It will be a hyperpolarizing stimulus. Okay. So this idea of depolarization and repolarization. So we said that depolarization, this is how we are decreasing the polarity. So we're taking the inside of the cell, which is very negative, and we're trying to move it closer to zero and make it more positive, right? So in order to make it more positive, you have to add positive charge, right? So in other words, we're adding sodium, right? So we're bringing sodium in, sodium flows in. This increases the membrane voltage. Whenever you repolarize, or during repolarization, you are reestablishing the polarity. Okay, so in other words, you're taking positive charge and you're taking it out of the cell. By taking it out of the cell, you make it more negative, and that's where we started, right? We started very negative. So if you look here, whenever we're moving up, this is depolarization, right? We're moving up, depolarization, number 2. Whenever we fall back down, number 3, this is repolarization. We're reestablishing the polarity. We're making it more negative again, okay? And so this is our action potential, and we'll talk about this in more detail in just a little bit. Right now, you can also call our depolarizing and our hyperpolarizing stimuli that we mentioned a second ago. You can call these EPSPs, or you can call them IPSPs. All right. So what this stands for is excitatory or inhibitory postsynaptic potentials. Right. So EPSP and IPSP. If you are excitatory, you are depolarizing, you're taking the membrane closer to threshold. Okay, you're moving it closer to zero, closer to that negative 55 threshold. So we're here at negative 70 and we move up. We became more positive. So this is an example of an EPSP. If we're at resting potential and we move down, we become more negative. This is an inhibitory postsynaptic potential. Right. So you can see here that we move more positive for our EPSPs. Whenever we repolarize, we reestablish the polarity, so we go back to resting potential, and then we have our IPSPs. Now for IPSPs, if we're making it more negative, you can add a negative charge, or you can get rid of positive charge, right? So you could have potassium leave the cell, that's positive charge leaving, or you could have chlorine entering the cell, that's negative charge entering the cell. Okay, so our EPSPs. So you can see here, all of these green waves here, these are examples of excitatory postsynaptic potentials. We're becoming more positive, right? We're moving closer to a positive value, right? So you can see the same thing here if you watch the animation, if you have access to the PowerPoint. And then IPSP is exactly the same thing, but completely opposite. So now we're moving more negative, so we are coming down, we're becoming more negative, so this is an inhibitory postsynaptic potential, right? Now, our postsynaptic potentials can do one important thing. They can summate All right, and what this means is this term summate or summation. If you take the sum of something in math, you're just adding the two things together, right? The sum of 2 and 2 is 4. So if we summate our postsynaptic potentials, we get some net result, okay? And specifically, the net result that we care about is at the axon hillock, okay? So we've mentioned the axon hillock, and we'll look at it again in a second, but this kind of purple-pink region that you see here, this is the axon hillock. Sometimes it's called the initial segment, sometimes it's called the trigger zone as well, all right? So here's the body, here's the axon, here are the dendrites, axon terminals. This region here, this is the axon hillock. The membrane of the cell has to be at -55 at this region in order for you to get an action potential. If you don't reach threshold here, then you don't get an action potential. All right. So there are two types of summations that we'll look at. All right, and both of these summations at this initial segment or at this hillock, they're trying to get us to our threshold. All right, our threshold potential, which we said is somewhere around -55. All right. At this axon hillock, at this initial segment, there's a high concentration of sodium channels. Sodium channels will open, allow a rush of sodium into the cell, make it more positive, and this is the first phase of our action potential known as depolarization. All right. So here you can see again, depolarizing, we're moving up, becoming more positive. IPSPs, we're moving down. All right. So the way we summate, and I'm going to skip this for a second and come back, we can summate in two ways. All right. One is called spatial, and one is called temporal summation. All right. So this first one of spatial summation. If you think of the term spatial, think spaced out. All right. So one neuron, this one that you see here, it is receiving multiple different postsynaptic potentials that are either excitatory or inhibitory. It's receiving multiple across different areas on the postsynaptic cell's membrane. Right. So anywhere where you see red, these are representing IPSPs. Anywhere where you see green, these are representing EPSPs. All right. So you take all of these green positive EPSPs and all of these red negative IPSPs, and you add them together, right? And you summate them together. And what you get is you get some net result. And where you care about this is, again, at the axon hillock, right? So we're caring about it right at this region here. If the membrane happens to be at -55 in that region, you get your action potential. If it's not, then you don't. All right. So an action potential is different than the graded potentials that we looked at. Okay. So the graded potentials include those EPSPs and IPSPs. You can have a very large EPSP, you could have a small EPSP, large IPSP, small IPSP. Action potentials, there's only one type of action potential. Okay. It does not vary in its magnitude, it does not vary in its duration. There's just an action potential. So there's no large action potential and there's no small action potential like you get with graded potentials. All right. Okay, so there's spatial summation. So you can see multiple different postsynaptic potentials arriving along different parts of a neuron. And then you have temporal summation, right? So temporal summation is where you have a very rapid delivery of postsynaptic potentials along a single presynaptic neuron, all right? So you could just pick one of these presynaptic neurons, all of these around here, and just pick one of them. And so there would be an action potential coming in this way, or I'm sorry, a graded potential coming in, and then another one, and another one. Some may be positive, some may be negative. And you add all of them together, and again, you either get the threshold or you don't get the threshold. If you're at threshold at the hillock, you get an action potential. If you're not at threshold at the hillock, then you don't get an action potential. All right? So again, temporal summation, you have multiple arriving along a single point of a neuron. Okay, so here you can see how we summate. So we're either summating temporally, spatially. There could be no summation, or there can be cancellation of different postsynaptic potentials. All right. So here we have our threshold, right? So if we look at our threshold potential, this is our -55 or -50 or so. If you have one, and a DS stands for depolarizing stimulus, HS stands for hyperpolarizing. So if you have one depolarizing stimulus and you return back to threshold, or I'm sorry, you turn back to resting potential, and then you have another one, those didn't summate, right? So you don't move any closer to threshold as a result of the second one, okay? If we temporally summate, so we summate along the same point of a neuron, you have one EPSP come in, so one depolarizing stimulus, and then next to it you have another one, and this got you up to your threshold, so you get your action potential, okay? or at least you get a depolarizing stimulus that takes you far above threshold, which could lead to an action potential if it's at the hillock. You could also have spatial summation, in which you have one depolarizing stimulus, another depolarizing stimulus, and then another one, and that third one combines with the previous two, and you get up to threshold, and so you're supra threshold, right? So you're above threshold, which again, you may or may not get an action potential. And then you can have cancellation. So you could have one EPSP, you could have that followed by one hyperpolarizing stimulus or an IPSP, and those two cancel each other out, right? So obviously if you have a positive 10 millivolt EPSP, and then following that with a negative 10 millivolt IPSP, positive 10 and negative 10 are going to cancel each other out, and it's 0, right? So it's like nothing happened. Okay, so you can have this this cancellation. And so really what you're concerned about, or what you should be concerned about and thinking of, is what are the different types of postsynaptic potentials? What are these different types of graded potentials? And again, it's kind of confusing because there's different names for the same thing, right? So there's excitatory and depolarizing, all these different terms. But the idea to keep in mind is that we're trying to summate them, we're trying to add them together, we're trying to get to threshold of negative 55, and specifically we're trying to get to threshold at the axon hillock. If you can get to threshold at the hillock, you get your action potential. If you cannot get to threshold at the hillock, then you will not get your action potential. Okay? All right. So if we continue looking here, we know that our action potential is a result of supra-threshold stimuli. Okay? So we have different phases here, all right, of our action potential, and as a result of these different phases, they're going to all combine, and we're going to get some net result. All right. So what we have first is we have a resting potential. Right. So we've already mentioned this. We have a negative 70 millivolt resting potential. Now, if you note that it's very negative, right? The reason it's negative is because we have potassium leak channels. Okay. And potassium will leak out of these channels. And we said earlier that if the membrane is permeable to an ion, it tries to make the membrane potential closer to its own equilibrium potential. And what was the equilibrium potential for potassium? The equilibrium potential for potassium was somewhere around negative 89 or so, right? So negative 70 is kind of close to negative 89. So that's why it's closer to the negative 70 than it is to, or the negative 89 than it is to the equilibrium potential of sodium, which is somewhere around positive 60, 61, something like that. Okay. So you're resting. You then have some type of stimulus arrive, and this stimulus is going to be a graded potential. It's going to be an EPSP, an IPSP, or some combination of the two. You summate them, and again, if you get to threshold, you get your action potential, and if you don't, then you don't get your action potential. All right. So the first phase of the action potential would then be our rising, or what we'll call our depolarizing phase. All right. Now, when we talk about these different phases, it's important for you to understand, one, the order of them. Okay, so which one occurs first, second, third, so on and so forth. Which ion is involved, and thus which ion channel is involved. Which direction the ion moves. So in other words, does it move in the cell or out of the cell? And what does that do to the membrane potential? So does it make it more positive or more negative, right? So the order of the different phases, what ion is involved, and thus the ion channel, which direction that ion moves, in or out of the cell, and then what does that do to the membrane potential? All right, so the first phase is depolarizing. If we're depolarizing, we're taking away our polarity. So that is this region here in which we have our rising phase, okay? All right, so you have different amounts of permeability to different ions at different points, right? So during the resting phase, we said that we have a high potassium permeability, so you see potassium is able to leak out. We then have a stimulus, right? And so we begin the process of opening our sodium channels, and then during rising, we have sodium channels open, and you have a rush of sodium in, and so this causes our membrane potential to shoot up and become more and more positive, okay? All right, after our depolarization phase, we then have the closing of sodium channels, and then we have the opening of potassium channels, right? And again, what type of channels are these? Yes, they're ion channels, but how are they gated? These are voltage gated channels, right? So in other words, they're opening in response to a change in the membrane voltage. So we reach our peak up here somewhere around positive 30 or so, and that positive 30 is the stimulus for our voltage-gated potassium channels to open, all right, those ion channels to open. When this happens, we have potassium channels open. When you open potassium channels, you now increase potassium permeability. Potassium flows from high concentration to low concentration out of the cell, down its concentration gradient, taking with it its positive charge, and so we see a drop in the membrane potential. So we go from positive 30, and we begin dropping all the way back down somewhere to negative 75, negative 76, something like that. Okay. Now, you note that, and if we look here, here's our falling phase of this repolarization. So note that we have our potassium channels are closed. They're technically inactive, and we'll talk about that in a second. But we have our potassium voltage channels open, right? This K leak, this is the leak channels that are always open, right? But we have the potassium channels open, so you have a rush of potassium leave the cell. And when it does, it leaves and it takes with it its positive charge, making the membrane more negative. Okay. Now, this hyperpolarization phase, all right, what this reflects is the slow nature of potassium channels to close, right? So potassium channels are very slow to close. They respond slowly. And so this allows a little bit of additional and extra potassium to leak out of the cell. And so if that happens, you make the membrane a little bit more negative than it previously was. All right. So you have this undershoot. We were at negative 70 and we overshot it, and now we're at negative 75 or negative 76, something like that. Right. So this is referred to as the hyperpolarization, sometimes called the after hyperpolarization phase. Okay. And then you slowly increase the membrane potential. You get back up to resting, and then you have the process start over again. Okay. All right. Now, what is unique about specifically our sodium channels are the different states of the sodium channels. All right, so the sodium channels exist in different forms. All right, and there's three general ones. The sodium channels can be open, they can be closed, or they can be inactive. All right, so there are two different gates, if you will, for a sodium channel. There's what's called an activation gate and an inactivation gate. When the activation gate is closed, all right, the channel is said to be closed. All right, so the activation gate is closed, the channel is said to be closed. Sodium is not able to enter. All right, you then get the necessary and appropriate stimulus, so it becomes more positive. Right, you see here we're very negative. Here we becoming more positive, and our activation gate begins to open. All right. So we're in this opening phase. We're still sub-threshold because we're not at threshold yet, but by the time it opens, we are past threshold, and our sodium channel is said to be in an open state. All right. If a sodium channel is open, then sodium is able to flow through it from high concentration to low concentration and move down its concentration gradient and make the membrane more positive. Right. So this is the depolarizing phase. Now, after this happens, once we reach that positive 30 or so, our channel does not technically close, but it becomes inactive, right? So we said that we have two different gates. One was this activation gate that you see here in yellow. You also have this inactivation gate. The inactivation gate is on the cytoplasmic side of the cell, and it will swing shut. So we close the inactivation gate. Now the channel is said to be in an inactive state, right? And during an inactive state of the sodium channel will not reopen for anything, right? So in other words, the inactivation gate is not dependent on voltage, all right? So any other stimulus that arrives, any other thing that comes and causes the inside of the membrane to become more positive, it's not going to influence this inactivation gate, so it's going to stay closed, and so sodium is not able to enter. This is a phase known as the absolute refractory period, all right? What this absolute refractory period is a time during the action potential in which the membrane is unable to respond to another stimulus. No matter how large, no matter how great, if this inactivation gate is closed, the sodium channel cannot respond. It's only when this inactivation gate opens and our activation gate then closes can we then respond to another future stimulus and then have it open again. Okay. So here you can see, we'll talk about the different phases. So repolarization, what's happening with our sodium channels, so and the potassium channels. So in repolarization, we said that the potassium channels are open, potassium is flowing out. This is the absolute refractory period that we just mentioned. Okay. During that hyperpolarization, what you see is this phase way down here, and if we go back, it's this phase way down here. That's the after hyperpolarization phase. And this phase, our inactivation gate of the sodium channel, right? So this one here has begun to open. So we look at this phase here. So we're at this state right here. So it's open, but the activation gate then closes. Okay. So what this means, and remember the activation gate, it can respond to a change in stimulus. And so this means that we're able to respond to another stimulus, so we can have another action potential. However, in order to get the action potential, we have to have a stimulus that is greater than the previous stimulus, right? So think about why that would be the case. Why would and this is known as the relative refractory period. Why do we need a larger stimulus than before to get the same action potential, right? We said the action potential is the same magnitude, same length, same duration, but the stimulus to get there has to be larger. Well, if you recall, during hyperpolarization, we went to be more negative than we previously were, right? So we were at negative 70, and now we're saying negative 75, right? So what's the difference between negative 70 and negative 55? Well, that's just 15 millivolts, right? We'll say now during this hyperpolarization, instead of being at negative 70, we're at negative 75. What's the difference between negative 75 and negative 55? That's 20 millivolts. So we went from 15 to 20 millivolts. So what this means is that during after hyperpolarization, you can still respond to a stimulus, you can still have another action potential, but you have to have a larger stimulus than the previous one, right? And it's because you're more negative. You're in this hyperpolarized state, okay? Then after hyperpolarization, you slowly return back to resting membrane potential, and a normal stimulus would able to get you up to threshold and then get you to have your action potential. All right, so you can see this here. So the red or the pink, this is representing the absolute refractory period, and our blue is representing our relative refractory period, right? So during this phase, the inactivation gate is closed, or during this repolarization here, you cannot respond to another stimulus. You also can't respond to another one during depolarization because the activation gate is already open, right? So in order to get an action potential, you have to open the activation gate. But if they're already all open, there's nothing to open, right? If there's a door open and somebody asks you to open the door, you say you can't open the door because the door is already open, right? So that's the idea here. However, during this relative refractory period, our inactivation gate opens, but the activation gate closes, and so you're able to respond to a stimulus. But remember, we're more negative and we're coming down, and so it's going to take a larger stimulus to get us up to threshold. Okay. All right, we're going to stop here and we're going to pick up with our next part in the next video, or the next part.